And it's my pleasure to introduce Julia Joung, who's another member of the Zhang lab. She'll be talking to you about the basics of CRISPR, genome-scale CRISPR-Cas9 screening. Julia actually published a Nature Protocols paper in really the past couple weeks. It came out, right? Like about a month ago?
Yeah.
Yeah, so she has the up-to-date, freshest knowledge, and she's going to be sharing that with you right now. Thanks, Julia.
Thank you. Good afternoon, everyone. So my name is Julia, and today I will be continuing our workshop with a more focused session on genome-scale CRISPR-Cas9 screening. So forward genetic screens are very powerful tools for mapping specific genetic perturbations to a phenotype of interest. Traditionally, this was done by chemically mutagenizing flies, producing many different mutations in different genes, and then selecting the flies with the interesting phenotypes such as white eyes or wingless, and then going back and figuring out which genes had mutations that produced that particular phenotype. Even though this contributed to a lot of our developmental genetic knowledge, it was not very high throughput. And more recently, with the development of shRNA screening, we were able to more scale it up to the genome scale level. shRNA knockdowns have been used to exploit the endogenous RNAi machinery to knock down base pair complementary mRNAs. And because it is easy to sequence the shRNAs and map them back to the genes that were knocked down, shRNAs have been developed for genome scale screening and used to study a variety of biological processes. However, shRNA screens have been hampered by the fact that many shRNAs have high off-target activity and low efficacy. Recently, the CRISPR-Cas9 system has been developed for gene knockout. As Nina mentioned earlier, the Cas9 system, when guided to a specific gene of interest, can produce a double-stranded break that is most likely repaired by NHEJ, which is very error-prone and creates an indel that then leads to a premature stop codon, which will deplete the target mRNA through nonsense-mediated decay and as well as deplete the functional protein. Cas9 has also been used as a catalytically inactive enzyme that can activate transcription. In our lab, we've developed a Cas9 activator that involves combining 3 different activation domains using the MS2 hairpins on the sgRNAs to then transcriptionally upregulate target genes. Cas9 has also been used for gene knockdown by fusion with the CRAB inhibition domain. And today I will be focusing mostly on CRISPR-Cas9 knockout and activation screening. Because CRISPR-Cas9 has much lower off-target activity than shRNAs, it has been used for a very wide range of screening applications, from drug and toxin resistance to gene essentiality, as well as more recently used for studying non-coding elements. And CRISPR-Cas9 screening can be used to study any biological process with a screenable phenotype. So what do I mean by screenable phenotype? There are 3 different types of screening selections: positive selection, naive selection, and marker gene selection. So hypothetically, if a guide RNA marked in blue produces a screening phenotype that we want, in a positive selection screen, the blue guide RNAs provide a proliferative advantage to the cells. So at the end of the screen, the cells with the blue guide RNA have grown faster than the rest of the cells. And after deep sequencing, we will see that the blue guide RNAs have been enriched. The negative selection screens are the direct opposite of that. So cells with the blue guide RNA die off more quickly than the rest of the cells, and at the end of the screen, the blue guide RNA cells will be depleted from the screen. Marker gene selection screens are a lot more versatile in that if we have a cell line that has a GFP reporter for a marker gene of interest, and in our screening selection, we want to enrich for the any guide RNAs that can produce a certain gene expression signature, we can use FACS sorting to select out the guides that produce that green marker, either using an endogenous reporter or antibody selection. So today I will be giving a very general overview of how to perform a CRISPR-Cas9 knockout and activation screening. The fine details are in our recently published Nature Protocols paper. So today I will be focusing on the most important points to consider when screening. So starting with library construction, we have provided several different libraries on Addgene. And these genome-scale libraries include guide RNAs that target either the human or the mouse genomes. For the gecko knockout libraries, we have provided this library in a 1-vector or 2-vector system. And for the Cas9 activation libraries, we have it in a 2-vector or a 3-vector system. For library construction, there are times when we want to screen for a smaller targeted set of genes. And this is most useful when we are only concerned with a subset of genes. For example, if we only want to target genes that are druggable or kinases, for example. And by reducing the number of genes that are targeted in our library, we also reduce the number of cells that are required for screening. And I will go into the number of cells that are required for screening in a few slides. And so in our protocol, we provided a script for isolating the subset of the sgRNAs from any of our genome-scale libraries. In addition, we have also provided a custom library design script. This is useful for designing, for example, a library that targets all introns, all enhancers, etc. And starting with identifying a genomic region of interest, we then select a certain number of guides that our script focuses on minimizing off-target activity as well as maximizing on-target activity, and then selects for guides that are easier to synthesize based on a number of criteria. In our custom library design script, we recommend to include multiple guides per target, and it is very important to include non-targeting guides in the screen, especially when you are setting up the screen for the first time. It's useful to have those non-targeting guides so you know whether or not your screen is producing a lot of false positives, for example, or if your screening parameters still need optimization. For either the targeted or the full custom library, we provide a custom library cloning protocol that involves PCR amplifying the synthesized oligonucleotides and then Gibson assembling the oligos into a plasmid of interest. During this cloning step, we have a table in the protocol that tells the user how to maintain the representation of the guide RNA library by scaling up each of these reactions accordingly. The next step is super important. It is to verify that the sgRNA distribution in the library is fairly even. And we provide a 1-round PCR protocol for amplifying guide RNAs for NGS verification. We also provide a Python script to analyze the NGS data and determine if the guide RNA distribution is good enough to proceed based on several different criteria. So now that the library is complete, the next step is to package the library into Lentivirus. Most CRISPR-Cas9 genome-scale screens use Lentivirus because the Lentivirus integrates into the genome, which is very important for doing any sort of cell proliferative screens, because the guide RNA copies will get amplified as the cell divides. Our protocol includes both a lipofectamine and a PEI version for making lentivirus. PEI is very good for making large-scale lentiviral preps because it is very cheap and easy to make. AAVs, in addition, can also be used for non-dividing cells only. So next is the transduction and selection part of the screen. The transduction portion involves either spinfection or a mixing method. It entirely depends on your cell type of interest. Spinfection is more commonly used for fairly robust cell lines like HEK cells or cancer cell lines. And the mixing method I've been using personally for stem cell transduction or primary cells. And the titer portion of the screen is very important for determining the multiplicity of infection. The multiplicity of infection, or MOI, measures the number of cells that survives the selection versus the number of cells that survives without selection. And to determine the lentivirus titer, we will use many different amounts of lentivirus supernatant on a certain number of cells, and then calculate the MOI for each lentivirus supernatant volume condition. And the MOI is very important, and we set it to less than 0.3 for the guide RNA library, especially because we want to ensure that most cells in your pooled screen have only one guide RNA introduced. And after titering the lentivirus, we can scale up the transduction protocol accordingly. So how to scale up the transduction protocol? For example, for a library with 100,000 different guide RNAs, we recommend greater than 500 cells per guide RNA coverage. What this means is that during transduction, because we are transducing at an MOI of 0.3, only 30% of the cells are going to survive. So for transduction, you would have to start out with 167 million cells. And then during the screen, we continue to maintain this 500 cells per guide RNA coverage, which means that throughout the screen, every single time you passage a cell, you maintain 50 million cells. So this is why potentially doing a targeted screen for cell lines that are difficult to work with could be very helpful because of the number of cells that are required for a genome-scale screen. For knockout transduction, if you are using the two-vector system, you have to first transduce Cas9 at an MOI of less than 0.7, and then transduce the guide RNA library. Then we recommend starting the screening selection 7 days after the library transduction, because that is when the indel percentage saturates. And again, throughout the screen, maintain representation at over 500 cells per guide. And during screening, we recommend doing at least 2 bioreps and up to 4 bioreps for each screen, because at the end of the screen, we want to select candidate genes, and because the screen is a very noisy process, we will select candidate genes based on which genes were overlapping the multiple bioreps. So for transduction, if you're doing 4 bioreps and 100,000 guide RNAs per library, then you would have to start out with 167 million cells times 4. So it's a lot of cells. And then similarly for transcriptional activation, we first transduce the activator component, then the library. And for activation, we can start the screen 4 days after library transduction because the transcriptional activation is a lot quicker than indel saturation. Okay, so during the screening selection, there are several considerations. We want to choose screening parameters that maximize the difference between experimental and control conditions. For drug screening, this means picking the drug concentration at the IC50 value of the drug. And a lot of the times for a new screen, it's very helpful to set up the screen using a positive control from literature to determine the screening parameters, because the screen is a lot of work, and it can be a lot of work to try to optimize during each round of screening. And again, during screening, please maintain a coverage of over 500 cells per guide throughout. And if it's the first time that you're screening, it can be very helpful to collect multiple time points throughout the screen. Because, again, screening is a lot of work. So if at the end of the screen, you realize that you probably should have looked at an earlier time point or a later time point, you can come back to the cell pellet that you've collected and then harvest the genomic DNA from that. And throughout screening, it is very important to remain consistent. So no matter which protocol you choose to use, keep using that same protocol throughout the screen, especially between bioreps. Okay, and the next step is to harvest and analyze the screen. For screening analysis, in our lab, we have been using the rigor analysis tool, but there are many different screening analysis tools out there. The most common of which is also Magic, is very commonly used. And so I would also recommend using Magic, but rigor is the one that we've been using for our lab. Both of these screening analysis tools are very similar. First, you normalize the experimental condition guide counts to the control condition. So you have a set of guides that have been enriched or depleted, and you rank them based on their enrichment or depletion depending on which type of screen you're doing. And then for each gene, rigor will determine the statistical significance of having a certain guide ranking and then rank the genes, candidate genes accordingly. So at the end of the analysis, you will get a candidate list of genes. Then, based on these candidate lists of genes, we will take the average or overlap of the top genes from each biorep. Because the screen itself is a very noisy process. So if we see a gene that has been enriched across multiple bioreps, that gene is most likely a real hit. Finally, the last step of screening is the validation. Validation is just as important as the screen because the screening analysis produces a ranked list of genes. So we want to determine whether or not each gene is actually important for your phenotype of interest. During validation, we repeat the first step of the screen, basically, except this time, instead of using a pooled guide RNA library, we will produce each guide RNA that target the candidate genes individually. We have provided a protocol for cloning each guide RNA, packaging them into Lentivirus, and then the same transduction and selection protocols apply for validation. For validation of knockout, first, we extract genomic DNA from the validation cell lines. Then we provide a 2-round PCR protocol for amplifying the target site with Illumina adapters. In our validation for knockout, because there are so many different guide targets that we want to amplify, we recommend the 2-round PCR because this is more versatile and allows you to combine different barcodes with different initial first PCR primers. Then, after transducing the cell lines of interest with the guides, you want to verify that the guide RNA produces the screening phenotype that you were interested in, and as well as verify the indel. And for indel analysis, we provide a script that has several different options for calculating what the indel percentage is for the target gene. And of course, just because a guide RNA generates an indel at the gene of interest does not mean that the protein is actually knocked down. So we will always recommend performing a Western blot to make sure that the protein is knocked out. Okay, and then for validation of transcriptional activation, we have provided a homebrew protocol for rapid extraction of RNA followed by reverse transcription. This protocol is very similar and a much cheaper version of the commercial cells to CT protocol. It involves maybe roughly 15 minutes of prep work, and then the actual cell lysis and RNA harvest takes 8 minutes to get to the reverse transcription. So when you're doing a full 96-well plate for qPCR, is a very quick protocol to use, and I would definitely recommend it if you're doing large-scale qPCR. And then after RT, we recommend using Tachman qPCR. The Tachman qPCR is different from the Cyber qPCR in that Cyber qPCR relies on 2 primers, whereas the Tachman qPCR has 2 primers and a probe, so it has additional specificity for on-target. And similar to the knockout validation, we recommend verifying the screening phenotype, determining whether or not the guide RNA actually activated the target gene of interest, as well as performing a Western blot to see whether or not the protein was upregulated. So in summary, the most important screening considerations are first, to maintain coverage throughout the screen. We recommend 500 cells, more than 500 cells per guide RNA in the library. And This applies to the initial cloning phase of the screen, as well as down to the genomic harvest and PCR for NGS analysis of the screen. Throughout the screen, it's very important to include controls. These controls include non-targeting guide RNAs in the library to make sure that to determine how much noise there is in your screen. And it's also very important to include a condition without the screening selection pressure. And because we will normalize the experimental condition to the control condition when we're determining guide RNA enrichment. And finally, it is also important to include in your screen genes that are known to affect the phenotype as positive controls to determine if your screening parameters are appropriate for the screen. And finally, it is important to remain consistent throughout the screen. So using the same protocols for screening and validation, doing the same transduction protocol for determining the lentivirus titer and for screening, for example. And finally, I'd like to thank all the people who helped me with writing up this protocol, especially Silvana Koloman, who co-led the Nature Protocols paper. And with that, I'll take any questions.
I have a question. So I read this paper that says the nucleosomes impede the Cas9 access to DNA. Do you take into account where your gene of interest is or how it is packaged in the DNA in the cell line when you are screening or targeting it?
So the Gecko library does not do that. I don't know of any library that actually does that. It seems like you're saying that the nucleosomes will impede Cas9 binding, right?
Yeah.
It seems like from the literature, it does not affect the indel rate that much, especially after 7 days of selection.
Okay.
That's based on my understanding. And so I think most libraries are more concerned with potential off-target activity and the on-target efficiency of the guide RNAs rather than nucleosome blockade.
So you mean that even though if it's packaged tightly, you would still get the same result? What were you looking
It seems
out
like
for?
it doesn't make that much of a difference after 7 days
Okay.
of transduction, yeah.
Question over here. I have a rather practical question. So the oligos that you guys use for either amplifying the library or cloning the guides, do you always use a PLC purified or it's desalt? Because there's no need.
No, there's no need. I just use regular primers that I order from IDT.
Okay, and then the other question is, do you always add, for guides that doesn't start with a G, do you guys always add a G at the front to increase the production efficiency?
No, we do not do that. For the SAM library, there is actually no G in front of the guide. So that's something to take into account when you're analyzing the screen, is that none of the SAM guides in the library have a G appended. And we found while using SAM to screen for melanoma drug resistance that it doesn't seem to affect the efficacy of the guide for SAM.
Thank you for your talk. Which Cas9 did you use for this screen? And how would that be different using different Cas9s?
So in our lab, we have only used the regular Cas9, SP Cas9 for screening. We have not tried screening yet with ESP Cas9. And for the SAM libraries, we screen with the SAM dead Cas9.
Okay. Thank you.
Hi, I was just wondering, you mentioned you should have a positive control for your screen. So if you don't have one, let's say you're looking for a receptor that isn't, you don't know that if there's another secondary receptor. In that case, do you see in your screens that haven't worked certain like guide RNAs that sort of appear on top of your list that are not specific for your phenotype. Do you see anything like that?
I have not seen anything like that yet.
So you never got a screen that didn't work?
So
You know, they might be guide RNAs selected for proliferation. Those cells might be just surviving better than the others for certain reason, not because selection worked.
So usually we have the control non-treated condition, and those, by normalizing the experimental condition to the control condition, you can weed out all the guides that are just provide a proliferative advantage instead of like a proliferative advantage in the presence of your drug, for example. In our lab, we generally use a pretty robust model for demonstrating our screens, the melanoma BRAF resistance cell line. So we have not seen as much of that. Yeah.
Thank you.
A question here. Can you speak a little about the sort of the spectrum of fast dividing cells to slow dividing cells to non-dividing cells, and which one of these do you, is that something that you need to take into consideration? And if you go to a non-dividing cell, your experience with AAV?
I have not had, and personally, I have not had as much experience with that. Most people still use lentivirus just because it integrates in the genome and you do not have to deal with whether or not a cell is fast dividing or slow dividing. The only thing I can think about is the time point at which you end your screen might be different depending on how fast your cell is dividing if you're looking at a proliferative screen, or like how you will maintain your cells during the screen. That can matter. But we have not done the experiments to test out whether different delivery methods matter for different growth rates for cells.
Regarding about the geek core here, your library. So there is the one vector system and two vector system. So in terms of the this 1 and 2, for the specifically or selected, is any difference between these two, or it will be just one step or two step different?
The main thing that people have said for the one vector and two vector system is that, besides the fact that the two vector system is two steps, is that when you're packaging the lentivirus, if you're using a one vector system, because it's such a large plasmid, your lentivirus titer would be lower. That's the main difference that people have said.
Thanks.
Okay, well,
Yeah. Regarding the DNA, the last step you do DNA extraction for genomic sequencing, how much coverage do you suggest for the cells? How much coverage for the library coverage? Like you suggest 500 coverage for maintenance. For how much coverage for the DNA extraction?
for the DNA extraction, we also recommend 500 cells per guide.
Oh, that's
And we have provided a large-scale genomic purification prep for purifying purifying genomic DNA from millions of cells, as well as PCR purifying from that scale.
okay.
Yeah.
Thanks.
Can you hear me? Sorry, just a little more, one more question about the weeding out some of the things that you would expect or just purely due to proliferative stuff. Do you have a list of them? And B, do you have any plans to just take those out from the library because they're going to come up in every screen of a dividing cell?
So the proliferative genes depends on each cell type. Of course, there are a lot of the genes that are overlapping, but normally we just recommend doing a control to normalize to. So a control non-treated condition, so that when you normalize, the guide RNA that produces the proliferation will not be seen as enriched in your experimental condition. Of course, if cell number is an issue and you do see a lot of the proliferative guides in your final sequencing and you'd like to reduce that, there is also the possibility of doing a targeted screen, and we've provided a script for isolating the sgRNAs that target the rest of the genes.
Thank you.
Let's thank Julie again for her great talk. And